Thank you. Actually, before we do that, Dr. Hricak was supposed to join us as a second discussant, but she has a terrible cold, and she decided to save us all from being exposed to her today, so thank you, Heddy. But actually, she does have some slides, and in her place, Wolfgang will be able to present those.
Okay. It's just a brief, it will be very brief, just 3 slides that summarize her ideas about this, how imaging can be used in the drug development paradigm. And it's what she wanted to focus on is these 3 areas: patient selection, imaging as an in vivo companion diagnostic, dose finding, and really to help to change from the maximum tolerated dose, we talked about a lot this morning, to the biologically relevant dose, and then to assess treatment responses. And just a few examples to emphasize this. I showed an example how PARP inhibitors could potentially be used in the future to image this. This is that actually happened already at Memorial some years ago, where this ligand called FDHT that binds to the androgen receptor has been used to guide the dose of this anti-androgen called AON509 for phase 2 clinical trials. And you see on the y-axis the changes in the PET signal at different doses in this phase 1 study. And while the maximum tolerated dose was actually found to be 390, there was actually not much change in the PET signal after a dose of 120 had been reached, and therefore, as a kind of compromise, a dose of 240 was then selected for phase 2 clinical studies to have some safety buffer basically built in. There's similar results we obtained in an ongoing phase 1 study of an ER antagonist, where also changes in the imaging signal were used to define the dose for phase 2. And then finally, technical feasibility to develop this molecular diagnostic is no longer the limiting factor. And this is a table, and the purpose of the table is that you cannot read it. It is listing all the molecular imaging probes that are currently ongoing or have been recently completed MSK with INDs for molecular imaging agents that are radio-labeled. So this is certainly, there's a large toolbox available, and this can be also distributed to other sites once the techniques how to synthesize those traces have been automated. Then the challenges are validation and FDA approval for in vivo companion diagnostics. Infrastructure, I think, is how can we motivate sites to participate in those clinical trials, and then perhaps most importantly, actually, the people, the physicians with experience in oncology, but also in imaging and nuclear medicine that can not acquire those images, but can read those images in the appropriate way.